Good afternoon, children. In today's online class held in the morning, I have discussed this table of essential elements, their functions, and the hunger signs or deficiency symptoms. This table I'm informing you specially because some of you have missed the class, and you should know that this has to be studied from page 12.9 as discussed in the class today, and this continues on the next two pages because this includes the macro and micronutrients. So children, please learn these functions, number them point wise, and also the deficiency symptoms. So with this table we have come to an end of this ongoing chapter, that is mineral nutrition in plants. The rest of the notes are into your Google Classroom. They are already stored up. You can make up your notes, children, because some of you are still remaining with their notes which are pending. And let me tell you, here in these topics, especially after nitrogen fixation, all this is there: mechanism, biological nitrogen fixation, right? The steps, mechanism, etc. But from here I have given you extra, children. This is not in your book. So Azolla anabina symbiosis, azospirillum, and nostoc I have given you from outside. So please write it in your copies and stick to these. Then another reminder already given you in previous videos was a homework of the nitrogen cycle from page number 12.13. This has to be drawn and properly labeled in your registers, right? So with this we come to an end of this lesson, and we will start with our next topic just now with lesson number 11, Kingdom Fungi. In your textbooks this is given page number 2.39 is where it is. Kingdom Fungi under biological classification. So let us start this with the general characteristics. Kingdom fungi is also called as the kingdom of multicellular decomposers. See, fungi is a plural term and the singular we call as fungus, right? Why we call them as decomposers? Because fungi that live in the soil along with some bacteria, they break down the dead remains of plants and animals. That is various dead bodies, decaying twigs, leaves, etc. They are broken down. So let us see the general characteristics. These are achlorophyllous, that means they do not have the chlorophyll. Spore bearing, their asexual reproduction is done by formation of spores or propagules. And non-vascular organisms. Non-vascular means they do not have the conducting tissue as xylem and phloem. All right. Now, fungi, they form a large group of organisms with over 1 lakh known species till today, right? So these general characteristics, they are for all of those species that are concerned. Second is, cell wall is made up of fungal cellulose, also called chitin. The fungi store glycogen as the reserve food. Then they are usually filamentous or thread-like with somatic, that means vegetative bodies. All right. These are heterotrophic, and that is why they were separated from plants. If you remember the five kingdom classification given by Whitaker, right? Kingdom Monera, this I have given you earlier. Let me just remind you very quickly. Here, the five kingdom classification as given by Whitaker was into kingdom Monera, Protista, fungi, plantae, and animalia. So why was fungi given a separate kingdom? The only fact was that these are heterotrophic, right? They could not make their own food, and that is why they were separated from the plants. Fungi decompose organic material and recycle the inorganic and organic molecules in environment. And finally, they cause several diseases in animals, plants, and also humans. Right? So these are general characteristics already written. Some terms I would like to introduce. The first is mycology. Mycology is the study of fungi. Another term, fungal pathology. This is the study of disease causing fungi. So they are not the same, children. This is the overall study of fungi called mycology, but fungal pathology means only the study of disease causing fungi. Another word called as caprophilous fungi. This includes, this means those fungi which grow on cow dung, right? We call them as caprophilous, right? So let's talk about the habitat. Now, during rainy season especially, fungi can be seen growing on various food items, wood, shoes, leather, then clothes, pickles, jams, cow dung, etc. Right? So fungi, they can be seen usually in rainy seasons, right, into all these articles. Then, due to lack of chlorophyll, fungi cannot make their food and therefore are heterotrophs. Just now I told you why they were separated from the kingdom Plantae. All right. Then they usually grow as saprophytes or parasites, right? But there are various articles that they are seen in the rainy season, especially. So fungi, they are saprophytes, that is, they grow on dead organisms, or parasites, that is, living inside a living host, or symbionts, or that means that it has a relationship of mutual give and take with another partner. Fungi which are parasitic, they possess haustoria which penetrate the host cell and absorb nutrients. I'd like to just show you the haustoria at page number 2.40. This is the fungus which is penetrating the host cell, haustoria, to derive the nutrition. So this is one of the characteristics of the fungus. Coming to structure, the body of a fungus is thalloid, which means that it is not differentiated into root, stems, and leaves. All right. The thallus consists of long tubular branched filaments called hyphae. Now again, hyphae is a plural term. The singular is called hypha. So the hypha, they look like this. They are branched tubular structures like this, long, right? One is called as hypha, but plural of the hypha is hyphae. The mass of hyphae is called mycelium. Remember, children, this is one structure. Sometimes they are all entangled up into knot-like structure. Then that mass of hyphae is called mycelium. All right. Now, hypha, they can be of two types: septate, that is bearing septa or partitions, and aseptate, that is without the septa, right? Septate are the characteristic of higher fungi, whereas aseptate, they are found in the lower fungi. All right. Under septate, we can talk about three forms: monokaryotic, dikaryotic, and the multinucleate. So monokaryotic means they bear one nucleus, dikaryotic means two nuclei, and multi means many nuclei per cell. So you can imagine here, look into the diagram. This is the first A is aseptate. You cannot see any partition here, right? So this is aseptate. But the second is septate hypha. You can see various partitions. And in each partition, you can see there are vacuoles, there are nuclei, etc. So depending on how many nuclei are per cell, you call them as monokaryotic, right? Monokaryotic septa, or dikaryotic, or multinucleate. All right. Another term to learn is cenocytic, which means a multinucleate hypha, or a hypha that has many nuclei. Right. There's another note. The heading of the chapter says multicellular decomposers. Fungi, they are multicellular decomposers. But there is an exception, right? Yeast is a unicellular fungus. The baker's yeast, or Saccharomyces cerevisiae, that you studied in your previous classes, is unicellular. Then, coming to reproduction, reproduction is by three methods. In fungi, it's by three methods: vegetative, asexual, and sexual. In today's this video, we'll talk about only vegetative propagation, reproduction. So vegetative reproduction can be by these methods: fragmentation, fission, budding, sclerotia, or rhizomorphs. Right? I'll show them one by one in the diagram here. This is fragmentation. In this, what happens? The hypha is broken into some portions of fragments, and each portion or fragment under favorable condition develops into a new fungus. So this is seen in Rhizopus and Mucor, the bread mold. All right. So fragmentation, as I just said, is the method of reproduction, that is vegetative reproduction. In favorable conditions, the hypha breaks into fragments like Mucor and Rhizopus. Next comes fission. Fission means to divide. You can see here one cell divides into two. You can see the parent cell splitting into two daughter cells, right? And each develops a new individual. This is seen in yeast. The third comes as budding. This is budding. This is found in yeast once again. And in this, what happens? A small outgrowth appears on the vegetative cell, which gradually enlarges in size, right? And then the bud is cut off from the parent cell. Sometimes the bud may remain attached to parent cell for a long time, forming a pseudomycelium, or mostly it gets budded off and then this grows as a new individual. The fourth method is sclerotia. We also call it singular is sclerotium. These are more or less rounded. You can see cushion-like cylindrical structures, right? And these are formed in some fungi only. They're perinating bodies. That means they can escape the unfavorable conditions, extreme temperatures, very high or very low temperatures, etc. And when the favorable conditions arrive, they produce new mycelia. Example is claviceps. All right. And the last is rhizomorph. Rhizomorph, these are rope-like. As you can see, they look like a rope, twisted subterranean. That means they are found below the earth's surface, and they are masses of several hyphae with well-defined apical growing point. This TS is not to be done, children, so we will not do about these structures. I am only explaining the external structures. Now these rhizomorphs, they also pass over the unfavorable conditions in dormant stage, and when the favorable conditions arise, they give rise to a new mycelium. So as we saw here, these are 5 structures discussed under vegetative reproduction. The same I have discussed here in short notes. About fragmentation, the same examples I have taken, what I have discussed from the book. Fission, as I just now discussed, about yeast. Budding, also seen in yeast. Sclerotia, seen in claviceps, the rounded cushion-like structures in the fungi as perinating bodies, how they under favorable conditions grow to form a new mycelium. And finally, the rhizomorph, rope-like twisted subterranean masses of several interwoven. That means they kind of form a knot-like structure and in favorable conditions grow into new mycelium. So in this introductory class just now, children, I'll be stopping here. In my next video, I'm going to start with the other video on asexual reproduction. So these 2 pages, children, you must write under general characteristics, these technical terms, habitat, structure, then something about hypha, this flowchart on reproduction, and vegetative reproduction, which I have completed in this video, and I'm going to post the next headings in next video. That is, I'll be giving asexual reproduction in the next video. So that's all for just now. Thank you.